Okay, good morning, and I want to thank the organizer first for a wonderful meeting, and thank you for inviting me here for the second time. So those who know me may expect, or my work may expect, that we will talk about reconsolidation because Rita started with reconsolidation. I'm not going to talk about memory reconsolidation and addiction, but if you're interested, just on the left of this door, we have a poster showing that something in a way similar to what Rita showed, that a combination of pharmacological effect, in this case it's MDMA, ecstasy, with a kind of primitive reappraisal approach in animals. We're doing aversive punishment in this procedure. During the reconsolidation window, it reduces relapse to alcohol drinking that's in animals. So it's very interesting. You are welcome to look at it outside. But today I'm going to talk about something else. So in the lab we have several directions of studies. One of them is indeed a reconsolidation, prevention of relapse, but the other is to understand the transition from moderate alcohol drinking or drug use. In alcohol, well, yesterday some of us had some Barak and some other beverages. That was, I hope, a social event. And in some individuals, it may lead over time, of course, and extensive drinking to the development of alcohol use disorder. And we have been working in the lab for the last 12, 13 years on studying the factors, and it's mostly biological factors, although not only, as Shaoli said yesterday, it's a neuropsychosocial disorder, but mainly on biological factors that control this transition from moderate social drinking to alcohol use disorder and other drug use. And so this is taken from a review paper that we published almost a decade ago. Dorit Ron from the Gallo Center, who was my postdoc mentor, and myself. And this is reviewing the literature, and since then there are much more. You don't have to memorize all these molecules, it's fine. Just I want to make a point with it. So here's the point. These molecules in these different pathways and molecular pathways from many researchers in the alcohol field were almost entirely taken from studies on neurons or on bulk brain regions. And in the field of addiction, there is relatively too little study on another population of cells in the brain, which are glia cells. And we became interested in the role of myelin and white matter in alcohol use disorder, in the alcohol addiction. And so we all know, probably we learned in our bachelor degrees, that myelin is crucial for the conductivity of the electric current in the neurons. It's a fatty tissue, and it's produced by glia cells called oligodendrocytes. And this is not entirely accurate, because the oligodendrocytes actually they have this extension that is wrapping the neuron with the myelin. And over the last 15, maybe 20 years, the term adaptive myelination has become very popular, because it appears that not only that myelin affects conductivity of neurons and the behavior and any output of the brain, but also the behavior can affect myelin in turn, and this can lead to some cycle of changes in myelination that can change behavior. And the question that we asked in this study was whether myelin is involved in alcohol addiction, and we are not the first to ask this question, of course. Both in human studies and in animal studies, this question has been addressed previously. In humans, mostly well, there are some post-mortem studies. Most of the studies were done using MRI, like DTI. And DTI and MRI are very important, but they are not looking directly at myelin. They are looking at some proxies of changes in the white matter. So this is more of the macro picture. In animals, more studies were done directly. Still in animals, many studies were done using DTI, MRI, but more studies were done directly on myelin. Mostly they showed either general abnormality of oligodendrocytes, these cells that produce myelin, or a decrease in myelin, mainly in myelin-rich areas like the corpus callosum and other commissures that connect different brain regions. And we wanted to concentrate here to focus on the changes in myelin in the mesolimbic system, in the regions that we know that play a major role in addiction. And surprisingly, not many studies have done so. So we focused on the brain reward system, the nucleus accumbens mainly, and its connectivity with the prefrontal cortex, medial prefrontal cortex. And our hypothesis was that alcohol changes first affects the cells that produce myelin, these oligodendrocytes, and then this can lead to myelin abnormalities which change the behavior, and adaptive myelination, changing the behavior further affects myelin, and then we have this, we thought about a vicious cycle in the striatum or in the brain's reward system. And we expected to see reductions in myelin because that's what most studies have shown. So I'm preparing you to the surprise, okay? So the first thing was to train animals to drink alcohol. We have a lot of models doing that. These were mice. And to see what happens to myelin, that's the basic, or to myelin-related factors in the brain. That's the basic thing. And the model that we used was a very simple model. It's called intermittent access to 20% alcohol in 2-bottle choice. It's a very long name for a very simple procedure. The animals receive alcohol every other day. So they have 2 bottles, one with alcohol, 20% in water, no sugar, no any additional thing. And the other is with water. They have food all the time. And we take out the alcohol bottle every other day, so it's intermittent. And we train them for 12 weeks. Some of the groups were trained for 3 weeks or in over 6 weeks. Now, most of the previous studies that I told you about in animals did not reach this long-term alcohol drinking of 3 months, 12 weeks. Most of them used between 1 week to maximum of maybe 5 weeks. So these are, it's considered in rodents long-term, but it's not as long-term as what we did here. So we trained them for 12 weeks, and this is the escalation of drinking that we observed in the mice. And we looked at several factors, I will guide you through it, that are related to myelin. We started by looking at gene expression, genes that are related to myelin. And it appears, I admit, I didn't know a lot about myelin before. I worked closely on this project with Prof. Barak, my colleague from Tel Aviv University, who's the myelin guy in our department. And it appears that oligodendrocytes, the mature oligodendrocytes that produce myelin, the myelinating oligodendrocytes, are starting from OPCs, oligodendrocyte precursor or progenitor cells. These are the earlier version of them. They are not myelinating at this stage, and then they differentiate to oligodendrocytes but they are still not myelinating, and eventually after maturation they mature to oligodendrocyte myelinating cells. And each of these processes is characterized by changes in expression of different genes. So these are only examples. SOX10, for example, plays mostly a role in differentiation of OPCs, of the progenitor cells to pre-myelinating cells, and then MIRF is playing a role mostly in the maturation to myelinating cells, and MBP, myelin basic protein, is the protein that is already participates in the structure of myelin, of the myelin complex. So we looked at these 3 genes and some other genes. This is already published, so if you want to see, you can see more genes in the paper. And we saw that at the earliest stage, at 6 weeks, we saw an increase in SOX10, suggesting that there was some kind of a differentiation starting to go on. Later on, this was, we observed a mirror effect, so there was kind of a rebound effect, reduction in SOX10. But at 12 weeks, there was a significant increase, and this was replicated several times in our lab. This is the nucleus accumbens. We also observed very similar picture in the medial prefrontal cortex, an increase in MIRF that plays a role in maturation, and in MBP, the myelinating oligodendrocytes, increases in them after 12 weeks. So to summarize that, we showed that long-term memory, and this is only long-term memory. This was not seen in most previous studies because they didn't reach this long period. So long-term memory, alcohol consumption leads to increases in myelin-related genes. So these are mRNAs, and mRNAs is nice, but we usually want to see also the protein. So we chose to focus on MBP because that's in the myelinating cells, and we dissected the nucleus accumbens, and we observed that it's not only the mRNA, it's also the protein itself. MBP itself is increased after 12 weeks of alcohol drinking. So it's both the genes and the mRNAs, both the genes, mRNAs, and the protein itself. And so this is Western blot. We used another method of staining in the tissue itself. It's hard to see here with the light, but anyway, what you should see here is that there are more red dots. These are staining for MBP, more red dots in the ethanol group. And the interesting thing that we saw here is that it's not only more red in general, it's more of these dots, or what we called MBP events. And this suggested for us that it's not only a general increase in MBP, but increases in the bundles of neurons that are going through the nucleus accumbens. So to test more directly, a little bit more directly, this hypothesis, first we counted these numbers and we saw, I'm not showing it here, but when you're quantifying it, you see that there are more MBP events. But we also used another approach, which is using connectivity assays using MRI. So this was DTI. This is a nice picture from, I think, GPT. The proportions are not exactly, but it's nice. It took me a lot of time to get this picture. So this was an MRI study, and we scanned the brains of mice after 12 weeks of alcohol drinking. And we actually looked at the whole brain. I'm not showing it here. If Bumi was still here, Bumi Tsangan was still here, I would tell him that his human alcohol drinkers are a very good model for my alcohol-addicted mice, because he found that globally there was, if you recall from yesterday, globally there was a reduction in connectivity. We found the same thing in the mice. But here, in what I'm showing you here, we are focusing on the connectivity between the nucleus accumbens and the medial prefrontal cortex. And so these are at least some of the bundles that we observed before. And we showed that there was an increase in the number of tracts connecting the nucleus accumbens and the medial prefrontal cortex. So this suggests that long-term alcohol drinking also increases nucleus accumbens and medial prefrontal cortex tracts and connectivity. So what do we have so far? We showed that alcohol drinking increases genes that are related to myelin. They increase proteins that are related to myelin after 12 weeks. They increase the connectivity of these brain regions, the nucleus accumbens and the medial prefrontal cortex. But still I haven't showed you the holy grail, which is what happens to myelin itself. So The next stage was to look at myelin itself, and for that we used electron microscopy at the ultrastructure level to show actually what myelin itself, what happens to that. So we trained animals for 12 weeks in the same procedure, and these are the water drinking animals. So these are cross sections of of tissues from the nucleus accumbens, and what you see here, the darker part, is the myelin. So these are myelin shifts, and these are animals that drank ethanol, and I think that even by eye you can see that it's thicker. So when we quantified it, we showed that the myelin surrounding of the axons was thicker in alcohol drinking animals. That's what you see here. When we measured the inner diameter of the axon itself, we did not see any difference. That's this graph. And then in this kind of studies, people usually show G ratio, which is the ratio between the inner diameter and the outer diameter. So the outer versus the inner within this myelin. And when it's going downwards, it means that there is an increase in the thickness of myelin itself. So I hope that I convinced you, and this was all surprise for us. I mean, at this stage, not anymore, but that we have a lot of evidence showing that unlike what we expected, we have an increase in myelin in alcohol drinking animals in the nucleus accumbens. This is isolated to the nucleus accumbens. As I said, reduction in connectivity in other brain regions, but in the nucleus accumbens, it's not a myelin-rich area, but still there is myelin there. There was an increase in myelination. So long-term alcohol consumption leads to increase in myelin thickness in the nucleus accumbens. So I started by saying that alcohol may affect oligodendrocytes. So we looked also at the oligodendrocytes at the cellular levels, and I'm not going to guide you through all these different markers, but for the different development of oligodendrocytes from the progenitor cells to the mature myelinating cells, there are different markers, molecular markers that you can use. So I will try to guide you through a little bit more easily on this one. So at the 3-week time point, we did this study at 3 weeks, 6 weeks, and 12 weeks. At the 3-week time point, short-term alcohol drinking, we observed an increase in the markers that signify an increase in the OPCs, the precursor cells, and a reduction in mature oligodendrocytes. That after 3 weeks, and it makes sense with what other people showed after short-term alcohol drinking. There was a reduction in myelin. At 6 weeks, suddenly we have a flip in the process. So there was a reduction in the precursor cells, but an increase in the mature cells, starting to probably myelinate. And at 12 weeks, we didn't see the difference in the cells anymore. So while there is no difference at the cellular level, we know that at 12 weeks we have a difference in gene expression, in protein expression, and in myelin itself. And this means that the abnormalities in the development of the cells of the oligodendrocytes begins before the changes that we see in myelin, the increases that we see in myelin itself. It precedes these changes. Okay, so the final piece of data that I want to show you is perhaps the most important thing, because so far I showed you how alcohol affects myelination processes. And now I want to show you the other direction, how manipulating myelination processes can affect long-term alcohol drinking. So to show that it's not only that myelinating is changing due to some processes in the brain that we still don't know how exactly it does it. That's one of the things that we will study soon. But that these effects are indeed leading to adaptive myelination, changes in myelination that affect behavior. So for that, it's not easy to manipulate in a good way, in a precise way, myelin, because there are some manipulations that can destroy the myelin, but then what you get is something like multiple sclerosis, and the animals can't move and so on, so we can't really use those models. What we chose to use is clemastine. That's a drug, a medicine that is used also in animals. It's an antihistaminic drug, but on top of being an antihistaminic drug, it also was shown to enhance myelination. Specifically, it can be measured that it enhances the number of mature oligodendrocytes, and it increases MBP, this myelin basic protein, and also the myelin amount itself. In our study, we first validated that it does it in animals that are drinking alcohol, and we showed that animals that received a supplement of clemastine for, it was 7 weeks, showed an increase in MBP, in the myelin basic protein, and an increase in the oligodendrocytes. So this was just validation of the manipulation. And then we trained animals for 12 weeks to drink alcohol, like we did in all our models. And upon reaching this 12-week time point, we started them on a regimen of 7 weeks daily chronic injection of clemastine versus vehicle, versus placebo. And these are the 7 weeks of drinking while receiving the clemastine treatment. So we see that they start more or less the same point, the vehicle and the clemastine, but clemastine is further increasing, clemastine-treated animals are further increasing their alcohol drinking, and at this point, I'm reminding you there is an even more increased myelin level compared to the vehicle-treated animals. So this suggests that enhancement of myelination, and I can't say it entirely 100% that it's only the increases in myelination, because clemastine is a dirty drug. It's doing other things besides increasing myelination, but that's one of the things that it does also in our hands. Enhancement of myelination increases alcohol drinking. So to tie all this together, we show that early alcohol consumption leads to abnormalities in the oligodendrocytes, in the cells that produce myelin. We show that long-term voluntary alcohol drinking leads to hypermyelination, increases in the genes that produce myelin, in the genes that participate in myelin production, and eventually thickens myelin levels in the nucleus accumbens, and that enhancement of myelination leads to further escalation in alcohol drinking. So this vicious cycle of alcohol consumption, enhanced myelination, and then strengthening the maladaptive behavior and drinking more, we are stuck in this vicious cycle, and this is one of the escalating factors that leads to escalation from social drinking, I started by that, to excessive and out of control drinking. So what is my conclusion from that? I had a teacher during my bachelor studies who said that in psychiatry and psychology there is a norm, and the deviations, the disorders, are when there is too little of that, too more of that, or it is changed in a qualitative way. The quality of the behavior changes. So you can think about mood. When you have too little of it, it's depression. When you have too much of it, it's mania. And when you have an abnormal quality of mood changes, it can be psychosis, right? And I took this very interesting and inspiring view also to other fields. And when we think about myelin, it's the same thing. When you have too little of myelin, you might have disorders, motor disorders and cognitive disorders like MS. When you have too much of myelin, it might be not good too, like we see here in the nucleus accumbens. It can lead to enhanced behavior towards the drug-associated cues and towards the drug. And when it's changed in a qualitative way, that can be a disaster, not a disaster. So what we need is a good balance. What we need is to have the proper amount of myelin along the brain, and although it changes with experience. And as for the question that I started from, does it belong to the GO pathway that enhances alcohol or stop pathway? So myelin, at least in the nucleus accumbens, belongs to the GO pathway. It increases alcohol drinking. And I want to thank my lab. This is not an updated picture, but I can take a picture from here of some of the lab. They are here sitting. And my collaborators on this study. So first, the study was done by Dr. now Mirit Liran and Dr. Inbar Fisher from Boaz Barak lab, who led the study, but some other lab members participated, of course, and Yaniv Asaf helped us a lot with the MRI. And I want to thank you for listening. Thanks.